I think if you agree, we'll go ahead just to keep on time, and I'll excuse me. I can, I can introduce the next speaker, who is Paul Franks. If you can put up his details there. So Paul Franks is very well known, I think, to the audience here, the participants. But Paul has had a lifelong interest in precision medicine in diabetes and has published extensively on this. Paul is a professor of genetic epidemiology at Lund University, and he's also an adjunct professor in Harvard. And importantly for today, Paul, together with Steve Rich, has co-chaired this whole initiative from the start. Without him, I think we wouldn't be here today, in fact. So Paul's talk today is about the Precision Medicine and Diabetes Initiative and his vision and how he sees the future direction. So I think it'll be very helpful to people who have joined us today to hear about the whole direction of the program. So Paul, turning it over to you. Thank you very much.
Thanks, John. Thank you. Okay. Can you hear me clearly? Good. All right. So I just really want to thank everyone who's participating in the next 3 days. Obviously, the organising committee's put a lot of work into this, and John's led that. And congratulations, John, for pulling everything together. But of course, this meeting wouldn't be a meeting without the 800 plus participants who are viewing and all of the speakers. And so I'm so happy to be part of this and to see this going ahead. So, I think we understand that diabetes is a global problem. It's a problem that is emerging. In other words, the prevalence of type 2 diabetes is increasing to the extent where there will be probably about 500 million men, women, and children by the middle of the century with diabetes, many of whom won't know they have the disease. That's a problem because untreated diabetes, of course, can lead to serious complications that are life-threatening. The burden of diabetes is global. It is emerging more rapidly in some populations than others, largely because type 2 diabetes in particular is a lifestyle-related disease, and as traditional subsistence farming populations migrate towards industrialized lifestyles, which is true in many parts of the world, the prevalence of diabetes will continue to increase. Now, we know that we can intervene in people at high risk of diabetes with different types of interventions that will slow the progression to diabetes. This is, of course, well-known data from the Diabetes Prevention Program outcome study, which shows the 10-year data. The three different cumulative incidence curves are for lifestyle at the bottom, metformin in the middle, and placebo at the top, of which about 1,000 individuals were randomized to one of these three interventions. The fourth arm, triglycerin, was stopped early. At the end of the randomization phase, at 3.2 years, the risk reduction in progression to diabetes in the lifestyle arm was 58% compared with the control arm, and 32% risk reduction in the metformin arm compared with the control arm. And this really mapped very closely to the results published a few months earlier from the Finnish diabetes prevention study that also had a lifestyle intervention very similar in design that showed a 58% risk reduction compared with their control arm. And this, of course, was really heralded as very, very important news in the fight against the progression to diabetes. And it shouldn't be understated how important the DPP and the Finnish diabetes prevention study and the other trials around the world that followed that showed that the same types of interventions have comparable results in other populations. Very, very important findings. Nevertheless, one should also consider not only the reduction in risk, but also the extent to which the progression to diabetes was delayed. Because of course, it wasn't really prevented in many people. Many people who were at high risk in the trial went on to develop diabetes later. So we can't really say that diabetes was prevented with these interventions. And on average, the delay is about 36 months with lifestyle and 12 months with metformin. Now, this in itself raises a question, which is whether, first of all, that's sufficient to really put a dent in the epidemic of diabetes. And secondly, whether we can do better than that. Can we delay diabetes even longer, or even prevent the onset of diabetes, meaning that it would never happen in people at high risk? And that really is where my sort of journey with the concepts of precision medicine began, when I started to think about how we might be able to prevent diabetes better. Now, these are results that we published from the DPP a couple of years ago that show essentially what happens to individuals in terms of their weight trajectories within the 3 treatment arms if they did or did not achieve a 5% weight loss goal at 12 months. You can see at the bottom those individuals who did achieve the 5% weight loss goal at 12 months. And those people did very well on both metformin and lifestyle, and indeed also in the placebo control arm in terms of weight reduction over the following 15 years. The lines in the middle represent the overall population. Just ignore those for a moment and now focus on the lines at the top. And what this shows you is essentially those who did not achieve a 5% weight loss goal at 12 months. And with metformin and placebo, the fact that they didn't achieve those weight loss goals meant that they would essentially track along more or less at their baseline weight for the following 15 years. However, in those who did not achieve their 5% weight loss goal with lifestyle intervention, you can see that their weight gain was quite considerable. This is weight change in kilograms, so 2 kilograms or so, or about 5 pounds of additional weight was carried across maybe a decade after the 12-month weight loss goal was missed. Now, what we didn't do in this analysis is look to see what impact that additional weight had on other features of health, but it would not be surprising if that was not a healthy feature of in these individuals randomized to lifestyle. So from this, I think we can conclude that not everybody will do well with standard of care lifestyle interventions, and therefore there may be good reason to think about other ways of prescribing preventive and indeed treatment regimes in overall populations. Now, you've already seen this slide, which is just really to pay credit to the many people who have been involved from the outset in the organization of the Precision Medicine in Diabetes initiative. But what I also wanted to do is to say that we now have more than 200 clinicians and scientists from around the world engaging in this initiative. People who have their own clinical practices and very much are on the front lines with dealing with diabetes, and people with excellent scientific credentials, particularly as it relates to big data and precision medicine. We have people with expertise in monitoring and devices. And now I can't see the corner of my screen because of the Zoom window. Also in pharmacogenetics and other features of medicinal treatment. And then, of course, in different elements of lifestyle, which is particularly pertinent in prevention of diabetes. What I also wanted to point out is that, of course, people often think of genetics in the context of precision medicine. It is indeed an important biomarker for all kinds of reasons, but there are many other types of markers that are useful, clinical biomarkers and other molecular biomarkers, omics, if you will, are also very important in this context, and therefore is the focus of our initiative. And we could not do this without partnerships, and those partnerships are with different organizations that you've already been told about, but also partnerships with patients and the pharmaceutical industry. I do want to reiterate what has already been said, which is that this initiative was really, the match was struck on this initiative by Will Cefalo back in 2017. And he really set out a mandate for what we should try to achieve across a 10-year period of running the initiative with this mandate and vision statement, which is written into the consensus report. So I won't read it out right now for the sake of time. And what we started with was a sort of model, if you will, that we mapped out in a different paper, but we carried forwards to the initiative, which was really to understand the path from sort of understanding disease etiology through to clinical implementation of precision diabetes medicines. Now, of course, a great deal of emphasis is placed on these more research-orientated activities, these traditional activities related with understanding disease etiology, looking at the role of biomarkers in different contexts, and then integrating that information into algorithms that might be useful for the prediction of treatment response or prognostication. But there are also many other features on this path that need to be paid attention to and often aren't really in discussions around precision medicine. So we explicitly flag these as important features that will need to be navigated in the process of getting discovery through to clinical implementation. And I should say that this was drawn up 3 years ago now, and since then we've added other features to this path, including health disparity assessment, which is indeed a very important thing to consider as we develop precision medicines of any sort, because we have the opportunity to either widen or close the gap on health disparities, and without evaluating that, we will not know what we're doing. You've already been told a little bit about the first consensus report, which was published last year in Diabetes Care and Diabetologia. The purpose of this report, which you can download from the meeting website, it's open access, was really to sort of lay some framework out for what we mean by precision diabetes medicine. One of the things we distinguished was precision from personalized medicine. Often those two terms are used interchangeably, and that's fine, but for the sake of understanding what we were talking about, we mapped it out as such. And at this stage, I think it's arguable that when we talk about precision medicine, we really aren't talking about personalized medicine, particularly in complex traits like diabetes. And the reason for this is that we're at best getting down to subgroups of the population that have similar characteristics that may be treated differently from other subgroups, but not truly down to the level of the individual. Now, of course, we should aspire towards that. And Dr. Cefalo talked about the need to keep the patient at the center and individualize medicine, and that is all true. But in terms of the work that we're doing at the moment, the research and ultimately, hopefully the clinical translation, it's likely to be really trying to minimize error in decision making, which is, of course, related more to the concept of precision. Within the consensus report, we mapped out different areas that we felt were important. The starting point was precision diagnostics, and you can see that we described a process that one might go through to get to a precision diagnostic for diabetes. And we highlighted that they're likely to fall into either sort of probabilistic scores, which would be more sort of a percentile ranking of risk, let's say, versus subclassifications or categorization of risk, which is along the lines of some of the work that's been published in the last few years, probably most notably from Emma Ulquist and Leif Group in their diabetes stratification that Dr. Cefalo also talked about. So there are different approaches, and those approaches are likely to manifest in a wide variety of ways. We also defined the concept called precision therapeutics, under which there's both precision prevention, but also precision treatment. And precision prevention, of course, is likely to focus largely on lifestyle factors, precision nutrition being a key area where there's some great data coming out now, but also precision treatment, which is likely to focus more on pharmacological treatments, but could also include lifestyle and surgery. And then the third component of the precision therapeutics map is precision monitoring, which, of course, if you really want to know how anything is going, you're going to need to measure or monitor it. And so precision monitoring, which we'll hear about later in this meeting, was an additional key component. And then we touched on precision prognostics. And I have to say, we didn't go into great detail in the first consensus report about this, but we really wanted to highlight that at the end of the day, diabetes isn't what kills people, it's the complications of diabetes. And therefore, if you really want to improve the efficiency and optimize the process of treating or preventing diabetes, you will need to consider what the prognosis is likely to be. And so this is an additional area we place priority on. So that led to the first consensus report, as I've said. We're now working on the second consensus report, which will actually be a series of papers, also will be co-published in Diabetes Care and Diabetologia, ideally at the end of 2023, perhaps even a little bit later. But this is very much based on trying to systematically review and grade the evidence that's in the published literature, focusing on the most important research questions for the implementation of precision diabetes medicines over the next decade. And those questions are being derived by working groups formed from about 200 experts in these different fields from around the world. So what we've done so far is the first consensus report. This series of consensus reports for the second stage is underway. And this is all leading towards a public-private partnership research program, which is really meant to fill in the gaps that are identified in the second consensus report, trying to understand what needs to be done, what should be studied more, not merely the sort of discovery and the etiology and biomarker end of that spectrum I showed you earlier, but also the other features of translation to ensure that if good ideas around precision diabetes medicine are discovered, they're also appropriately translated into clinical practice. So I just wanted to end with this quote, which is one of my favorites, and it's a very old quote, but I think people often ask whether it's too early to be talking about precision diabetes medicine. But it's very hard to be doing anything interesting at the right time. And if you wait, you'll be too late. And so I do like this this quote, which is paraphrased as "If not now, then when? If not us, then who?" Thank you.
That's terrific, Paul. Thank you very much. And that was a wonderful quote there at the end, and also a very nice overview of where this whole initiative is going. I think maybe you've mentioned there that 200 people are involved now in the working group process that you just alluded to. Can you say a little bit more about that, Paul? Exactly where that's happening and how it's being coordinated?
Yeah, so this is a fairly large beast, I would say, that seems to be growing weekly. So we started with a core group of world leaders in precision diabetes medicine, and a large number of early and intermediate stage researchers who are aspiring to be leaders in the field. And we brought those people together to work in a standardized fashion to review and grade evidence that's in the published literature. And as I said a moment ago, the idea is really that we'd like to know what the foundation is for precision diabetes medicine today in order to be able to plan for what needs to be done in the future in terms of new research and clinical translation. And I think this is tremendously important because as much as I love the concept of precision medicine, it is prone to hyperbole. And I think we do really need to understand, objectively where we stand, and not merely what the great ideas are and possibilities in the future.
Terrific. So it just shows, I think what Will Zeffelu had also said. This is a long term. This is for the long haul. This is not a short term project. Clearly. Are there any other questions, Christine? Do you have anything to add?
No, I'm keen on seeing and being part of the process, and I think it is growing. Paul, I notice even in the chat you have people eagerly wanting to be involved, and I think with each session we're welcoming it, getting it more global, getting more stakeholders involved, and I'm enthusiastic about seeing how it evolves over the next several years.
And I could maybe add that I should have said earlier on, obviously we've been working closely with colleagues in India and China and other countries as well. I noticed I got a message earlier on that we're translating these sessions into Chinese, and there are 1,000 delegates from China logged on today, so which is fantastic to know. So this is really engaging people from everywhere. There's a question here. Sorry, I just see one here. This is a question from Steve Rich. The clustering that you, Paul, and Will Zeffelu mentioned from the studies in Sweden from Life Group, they're applied to a small subset And it's a question about how well does that cluster maybe work in other populations. I think there has been some work on that, Paul.
Yeah, there has indeed, and certainly there are a number of papers that have been published that have essentially reproduced the clusters in other populations. Although I have to say that the work that's been done in India on the clustering leads to a slightly different conclusion. But I won't go into that now because Steve Rich will be talking about that tomorrow. Or at least the session he's chairing will be discussed. So yes, on the one hand, the clusters themselves have been reproduced, which isn't really surprising because the way the clustering works is essentially using the correlation structure between the variables that are included in the model, and if the correlations between those variables are similar from one population to the other, you would expect those clusters to be reproducible. So I think it's particularly interesting when you start to look at clustering in populations that don't have the same relationships between these different variables, C-peptide and BMI, for example. So yes, the approach has been reproduced certainly in European ancestry populations with some distinctions from other ethnic groups. The other thing I'd like to say on this, which is super important, is that we've recently surveyed the amounts of research that's been done in GWAS studies over the last 15 years. And when you look at where that data comes from, the vast majority of it across all complex disease areas really is, of course, from European ancestry populations. And indeed, a very large chunk of that has come from the Nordic countries. And so you have a very homogeneous group of individuals contributing the majority of the data to many GWASs, particularly in diabetes and cardiovascular disease. And what we don't have is much data from some of the other large ethnic populations around the world, East Asians to some degree, but most other populations are not well studied, and that's for genetics. But I think it's really important that as we progress with this work on precision medicine, which of course is more than genetics, we do diversify, because what we learn will be channeled into translation pipelines. And if that really only works well in European ancestry populations, then that will not do service to, of course, the global population.
Okay, Paul, in the interest of time, we'll have to move ahead, and thank you for an excellent presentation. And we look forward to hearing more about that tomorrow, that last point tomorrow. I'll hand over to Dr. Leave to introduce the last speaker.
Thank you.